In today's video, we'll be studying spermatogenesis. And here we have the outline of all we'll be discussing. The first meiotic division, the second meiotic division, and spermiogenesis. Now, earlier we considered oogenesis, and we saw that in oogenesis, the process starts in fetal life or in prenatal life. However, it's good for us to take note of the fact that spermatogenesis actually starts at puberty. Okay, and it basically involves the various steps that lead to the formation of spermatozoa or sperm. Okay, now another thing for us to take note of as a difference between oogenesis and spermatogenesis is the fact that when we considered oogenesis, we saw that we had the formation of a secondary oocyte as well as polar bodies. So we had the first polar body forming in the first meiotic division and the second polar body forming at the end of the second meiotic division when fertilization takes place. So at the end of the day, what we have is just one ovum formed. Okay, however, for spermatogenesis, we have at the end 4 sperms. So each of the processes actually give rise to a sperm. Okay, so unlike in oogenesis where we have just one ovum forming at the end, here we have 4 sperms. Also, we saw that for oogenesis we had a very prolonged prophase 1. The cell was in the resting phase of decluttine, and then it was in this phase up until puberty, just before the onset of puberty. However, for spermatogenesis, we would go on to see that prophase 1 only lasts for a period of about 22 days. Okay, so these are some of the minor differences that we have comparing spermatogenesis to oogenesis. Alright, so first we have the primordial germ cells. Now, the primordial germ cells are these precursor cells. They are basically stem cells, meaning that they are undifferentiated. So, in the embryo, in this male embryo, we have a bunch of cells, and this bunch of cells mostly are stem cells and precursor cells that have the potential of forming other cells. Okay, so some of them have the potential of forming just a particular type of cell, some of them have the potential of forming multiple types of cell, and an example of a precursor cell is the primordial germ cell. Now the primordial germ cell is actually formed in the epiblast. Okay, now in the introductory video, the very first introductory video, when we considered the introduction to embryology, we saw that in the formation of the embryo, we have the formation of the bilaminar germ layer as well as the trilaminar germ layer. We also saw that for the bilaminar germ layer, what we have is the epiblast as well as the hypoblast. So these primordial germ cells are formed at the epiblast at the second week of embryonic life, and they move on to the yolk sac. Now, the yolk sac is basically a temporary structure that provides nourishment for the embryo. Now, this yolk sac is away from the developing gonads, so the primordial germ cells would have to move on to the developing gonads, and so they migrate to the developing gonads, which in this case, since we're considering a male, is the developing testis. So they migrate to the developing testis at about the 5th week of development in embryonic life. Now, when these primordial germ cells get to the testis, they differentiate to form spermatogonia. Okay, and as we would see, experience a phase of dormancy and inactivity up until we get to puberty. This is unlike oogenesis, as we saw earlier, where the oogonia would further proliferate and undergo differentiation to form the primary oocyte. in spermatogenesis, what we have is that we just have a differentiation from primordial germ cells to spermatogonia, and it stops there up until puberty. Okay, of course the spermatogonia is diploid, XY, because this is a male, so we have 22 pairs of autosomal or autosome chromosomes, and then a pair of allosomes, which are the sex chromosomes. So in the male, before the onset of puberty, okay, so before the onset of puberty, now we have this spermatogonia, and this spermatogonia, as earlier stated, they are diploid, okay, 46XY. Now what we have is that this spermatogonia proliferates, okay, so it proliferates and it multiplies, okay, so multiplication and proliferation takes place by mitosis. Now this is very important for us to take note of. For the proliferation in the males, the proliferation of spermatogonia in the male, here's what happens. We have what we call the type A spermatogonia as well as the type B spermatogonia. Now, take for example, we have just one spermatogonia right here. This spermatogonia would undergo mitosis in order to proliferate. Now, of the two cells, of the two daughter cells formed by mitosis, one would be a type A spermatogonia and another would be a type B spermatogonia. Now, the type A spermatogonia would not differentiate further. This type A spermatogonia would remain stem cells. Remember that these spermatogonia are still undifferentiated. Okay, they are still undifferentiated cells. So the type A spermatogonia would remain as stem cells, they would remain as spermatogonia, while the type B spermatogonia would go on to form the primary spermatocytes. Okay, so they would differentiate further into the primary spermatocytes. Just before the onset of puberty, these primary spermatocytes would actually undergo the first meiotic division. Okay, so the first meiotic division would occur, and we also have a prolonged prophase. So the prophase 1 lasts for about 22 days. So prophase 1 for about 22 days, and this is of course prolonged, but it is not as prolonged as we have in oogenesis, where the prophase 1 lasts from prenatal life to just before the onset of puberty. So this prophase 1 lasts for this duration, okay, and then after this period of time it undergoes the metaphase 1. Okay, undergoing metaphase 1 and of course anaphase, and it completes the first meiotic division. So now we have a secondary spermatocyte. Okay, so from a primary spermatocyte, from one primary spermatocyte, we have two secondary spermatocytes. Okay, of course, as earlier stated, this is unlike in the females where we have the first polar body and then one secondary oocyte. It's good for us to take note of the fact that all of these processes take place in the seminiferous tubules of the testis. Okay, now in the seminiferous tubules of the testis, we have the germinal cells, which are basically the spermatocytes, and then we have the non-germinal cells. Now the non-germinal cells are the Sertolic cells, and as we would go on to see, these Sertolic cells actually help in spermiogenesis. Okay, spermiogenesis is the conversion, it's a morphological conversion from spermatids to spermatozoa. So now we have the secondary spermatocytes. Okay, two secondary spermatocytes. Now these spermatocytes would begin the second meiotic division. So we have the second meiotic division. And it would begin the second meiotic division and, of course, complete this process to form spermatids. Now, these spermatids are haploid and it could be 23X or 23Y. Okay, so taking a look at this picture that we have here, we can see the entire process summarized in this picture. So here we have the spermatogonia, which is diploid, containing 46 pair of chromosomes XY, and then it differentiates into the primary spermatocyte, which is still diploid, 46 pair of chromosomes XY. Now, after the first meiotic division, we can see that it forms two secondary spermatocytes. These secondary spermatocytes are haploid. However, it's important for us to take note of the fact that although they are haploid, they actually are dual chromosomes. Now, the term chromosome can be used for two sister chromatids that are joined at a centromere, and it can also be used for a single chromatid. So a single chromatid such as this one could be referred to as a chromosome, and a dual chromosome such as this one is also referred to as a chromosome. Okay, so what we have here is not two chromosomes. Okay, what we have here is just one chromosome that contains two sister chromatids. Now, at the secondary spermatocyte level, what we have here, we have these spermatocytes containing 23 dual chromosomes. Okay, so they contain 23 dual chromosomes, not single chromosomes as we have here. Now, in the second meiotic division, what happens is that these 23 dual chromosomes are now split up. So what we now have here at this level of the spermatids is that we have 23 chromosomes, 23 single chromosomes. Okay, so initially here we had something like this, and now at the spermatid level, what we have would be something like this. So we still have two chromosomes here, although we have one chromatid each, and here we have two chromosomes that are made of two sister chromatids. Okay, so at the spermatid level, this is what we have. We can see that the division, we have this 23X giving us the spermatids that are X. So a fertilization, where if this 23X spermatids that go on to form the spermatozoa fertilize an ovum or an egg, they would lead to the development of a female embryo. Okay, and here we have the Y that would lead to the development of the male embryo. Now let's take a look at the process of spermiogenesis. At the end of the second meiotic division, what we have is spermatids. Okay, so we have spermatids at the end of the second meiotic division. However, these spermatids are not morphologically mature, so they would have to undergo the process of spermiogenesis. Spermiogenesis. Now, spermiogenesis is a morphological conversion of spermatids to spermatozoa. Now, this process is neither mitotic nor meiotic. Okay, it's simply a morphological change. Have here in this picture is the spermatid. Okay, so we can see the structure of the spermatid as we have it here. Now, this spermatid would go on to undergo morphological maturation to produce the spermatozoa that we have here, which is made up of the head piece, the neck, and then of course the tail. Now, the middle piece of this tail, in the tail we have the middle piece, of the tail, we have the principal piece, and we have the end piece. Now, the middle piece of the spermatozoa contains mitochondria, and this mitochondria is what produces the energy for the locomotion of sperm, okay, in the female reproductive tract. Also, at the head piece, we have the acrosome. The acrosome covers about 2/3 of the head piece, and it contains certain enzymes that help the penetration of the sperm into the zona pellucida of the ovum. Okay, so right here, what we have is a mature spermatozoa. Earlier stated, all of these processes take place in the seminiferous tubules. However, this sperm that has been produced is morphologically mature, but it becomes functionally mature in the epididymis. Okay, so the epididymis is also a convoluted structure, although not as convoluted as the seminiferous tubules. So the sperm, after being formed at the seminiferous tubules, is transported to the epididymis, where it is stored, and it also undergoes functional maturation. Very quickly, it's also important to take note of the fact that the Setolic cells, the setolic cells, which form the non-germinal part of the seminiferous tubule, actually help in the absorption of some of the cytoplasm. Okay, so for the spermatids to be converted into spermatozoa, some of the cytoplasm has to be absorbed. The nucleus has to become more compact, as we can see in this picture. Now, the setolic cells help in the absorption of some of this cytoplasm. On average, it takes about 74 days for spermatogonia to be converted into mature spermatozoa. Okay, and about 3 million sperm are produced daily. Now, taking a look at our outline, we have come to the end of this video. If you liked this video, please give it a thumbs up and don't forget to subscribe. Thank you for watching till the end. Bye.